Chapter 21 Biotechnology and Genetic Modification. 21.1 Biotechnology and Genetic Modification. Objectives again, they are very important. Go back to them after you're done studying. Biotechnology. It is when we use microorganisms to produce required substances. What do I mean by this? You know diabetes. Diabetes is a very common problem in nowadays humanity, and we need insulin to survive. Many people need insulin to survive, so this is a required substance. But how can we extract it? Simply, we know that each gene codes for a protein, and a gene is a segment of DNA. So what if we can take this gene and make this code produce the protein insulin, so we can take it and give it to the person with diabetes who cannot make his own insulin inside his body. This is a good idea, but how can we do this? We need a microorganism that has a DNA where we can put this gene in the DNA of this microorganism, and now if this microorganism has the code, it will make the protein. This is what biotechnologists do. They take the gene of insulin and place it in a bacteria. Accordingly, the gene will code for the protein insulin. So now the protein insulin is synthesized inside the bacterial cell. Now biotechnologists will extract the insulin, make it a medicine, and inject the person who needs the insulin with the insulin protein directly. And this will help with the body or the blood glucose level. So this is a way of using microorganism to produce a required substance. Also, we have another way, which is genetic modification. Changing the genetic material of an organism. How? By removing, changing, or inserting an individual gene or some individual genes from one organism to another. And this is what we call genetic engineering. So we have biotechnology, using microorganisms to make a required substance, and we have genetic modification or genetic engineering, where we can change in the sequence of a specific gene or do the changes that we need in DNA. So why do we use specifically bacteria? First, they are very small, easy to grow in the lab. Also, they reproduce very quickly. You can get a huge number in a very small time. Also, they are able to make complex chemical substances like the insulin from the example I mentioned. Also, they are cheaper to produce. When you produce or make bacteria reproduce, this is much cheaper than making any animal, for example, reproduce or plant. There are no ethical issues in the production of bacteria. Nobody will shout out, where are the rights of bacteria? So these are the 5 reasons why biotechnologists use mostly bacteria. Let's answer some questions. What is not a reason that bacteria are widely used in biotechnology? Not a reason. Make sure that you read the question carefully, please. It is, they are complex organisms, because they are not. They are simple organisms made of only one cell. Yes, they produce complex chemical substances, but they are simple organisms. Which statement correctly explains why bacteria are used in biotechnology? This is the opposite of the first question. So it is, bacteria can make complex molecules. I put those questions after each other so you can differentiate and that you can notice that reading the question carefully has half of the answer. Define the term genetic engineering. It's the same as genetic modification, changing the genetic material of an organism by removing, changing, or inserting individual genes from one organism to another. Outline why bacteria are useful in genetic engineering. Outline questions can be answered in bullet points. So there we go. All the things that we mentioned in the previous slides. Bacteria very small, reproduce easily in the laboratory, and they reproduce quickly. They can make complex substances cheaper, no ethical problems. All of the points that we mentioned before. Thank you, and I hope this video helps you study.